Okay. All right. So I couldn't remember the title that I gave Ariel, so I did my best to remember it, and clearly it is not the exact same. So today I'm going to give you oh wait, is this
That's my fault.
blocking? Okay.
Maybe.
Okay. Okay. So, hi, my name is Emily. I'm talking about context-dependent and cell-type specific effects of nutrient-stimulated hormones on the genetic risk for neuropsychiatric disorders. Um, and oh, my slide's not advancing. That's good. Okay, so I work in two labs. I'm at Yale. I'm going to be a fifth-year PhD student, and this is just one of my projects in one of the labs, which is the Brennan's lab. My alter ego when I'm not doing cognitive neuroscience. So I'm going to start out by telling you that neuropsychiatric disorders are heritable, and we can study them using genome-wide association. And so that means if we imagine the dark blue guy was a case and the light blue guy was a control, we can get their DNA, usually from blood or saliva or something like that, and we can walk through the entire genome and ask the question, basically do a series of t-tests as is the allele an A or a T, and the case significant or is having A allele significantly associated with being a case or control. And we can do that in mass over thousands and thousands of individuals. Here's an example of a GWAS that was published in 2019 of anorexia nervosa. And if we plot the chromosome on the x-axis and the significance of the association on the y-axis, we end up with this Manhattan plot where we see that any of the green diamonds are SNPs or variants that are significantly associated with being a case. And so that's really cool. And someone went and looked and did a whole meta-analysis about the different GWAS and psychiatric disorders that have been done. And what's really interesting is that you would think that the DNA probably gives us all the information we need to see if something's heritable, but we can see here that that's not true. So if we zoom in again on anorexia nervosa, this is the same Manhattan plot, just made really tiny. You see that actually there's 30% of the variance is missing. So we can say that GWAS explains about 14% of this observed phenotype. And if we look at twins, then we can recover another 58%, or well, 58 minus 14, whatever that is, 36, something like that. Anyways, that leaves us with about 30% that's missing, which is crazy because you would think that the DNA would tell us everything. And so you also might think that why would we have these SNPs if they're not doing anything? This seems evolutionarily counterintuitive. And so what I work on is called EQTL, or basically the hypothesis that maybe what SNPs are doing, they're not directly coding for protein, but they might be regulating expression of some distant target gene. And the reason that we apply this approach, especially for psychiatric disorders, is because if we look at these GWAS, we know that most of these SNPs lie in non-coding regions of the genome. So they're not directly coding for protein, but again, it doesn't really make sense that we keep these after generation after generation. They haven't been selected out. Maybe what they're doing instead of directly coding for proteins is regulating the expression of a target gene. And another interesting layer you can add to this is that maybe this relationship depends on the cell type that you're looking in. So for example, GABAergic versus glutamatergic neurons. And also there could be some interaction with the environment that your cells live in, right? Your cells aren't in like a static environment. Your neurons, I'm really interested in nutrition, are exposed to different levels of ghrelin, leptin, or Ozempic throughout the day. And maybe this influences this relationship between a SNP, a non-coding SNP, and some distant target gene. And in my lab, not my lab, in the lab that I am in, we study it. I don't, people always say their lab, but it's not mine. The lab that I am lucky to be in, we study this relationship between SNPs and genes using something called stem cell derived neurons. And this is where we take a stem cell. You can see on the left side, these are actually colonies of cells. These were taken from skin biopsies, and then you can put them in a really cool chemical soup of different types of transcription factors. You revert them back into their pluripotent stage, which is what you see on the left, and then you can differentiate them into whatever cell type you would like, which, as I mentioned, for me are excitatory and inhibitory neurons is what I make. And you can make these really pretty neurons. You can put different things in there that make them glow nice colors. So this is how you do it. And you can expose the neurons to different sorts of compounds. And so the one I'm showing you as an example here is a GLP-1 receptor agonist, which I use Ozempic. So I differentiate these neurons. Some of them were exposed to Ozempic and some of them weren't. And I'm showing you here, if we just look at the gene level, that we do see that there is differential activity of genes based on exposure to Ozempic. So this is plotting on the x-axis, you basically have your effect size, and on the y-axis, you have the significance, and anything that's highlighted in red is significant, and the ones that are annotated are significant at an FDR, sorry, red is significant after being FDR corrected at 0.05, which is what we usually use instead of Bonferroni significance. This is all to say, if we look at just the relationship between Ozempic and a target gene, we do see some sort of phenotype that comes up, which is cool. So because it's only 5 minutes, I'm going to tell you the next thing I'm working on now is investigating this blue part, which is the relationship between the SNP to the gene. And we do this by doing a whole bunch of molecular biology that I never knew how to do before my PhD, but you can clone stuff into vectors and use viruses to put them into your cells. And it's this whole headache of a process. But basically, using all this stuff, you make up what they call a library of different SNPs. So you could put SNPs from anorexia, which I did, type 1 diabetes, type 2 diabetes, all into these different plasmids. And then you give these plasmids, these things into your cells. And then later we're going to be able to look at the ratio of DNA, so what you get in a GWAS, but also then you compare it to the ratio of RNA, which is actually getting transcribed, which is how you would tell the relationship between the DNA of the SNP and what's actually happening to the genes downstream. And that is what I'm looking for. Okay, sounds good. Thank you.
Thanks, Emily. Next up, Morgan. And Junbom Kwon on deck for the following talk. Great job, Emily. Very rapid speaking there. Morgan? Are you there?
Is Morgan here? Yep. So share your slides.
You're up. Talk about sleep renormalizes learning-induced changes in cortical excitability. And as a reminder, next up after that is Junbom Kwon, who I think is on Zoom somewhere. Oh, you're here. Hey. All right. Don't forget to share your screen.
I am working on it. Sorry. No. How do you share it? Like slides and not the presentation?
You should be able to just share slides, and then usually there's like a swap button.
There you go.
Sorry. Okay. Oh.
All right.
I could have pulled the notes up on my phone. Sorry. Took a photo just in case. Okay. Sorry, technical difficulties. I'm Morgan. I am a project coordinator at Brown Health right now, and I'm going to talk to you about some of the work that we just finished up. So it's about how sleep renormalizes learning-induced changes in cortical excitability. And this work, was mostly done in the Somnoro lab at the University of Massachusetts Amherst. So the dominant framework right now for sleep and memory centers on non-REM oscillatory coupling. So things like slow oscillations, spindles, and hippocampal ripples. So this complementary theory is called the synaptic homeostasis hypothesis. And it proposes that learning during sleep imposes a net increase in synaptic strength that eventually would lead you to the cap of like learning and further encoding. So the thought is that sleep will resolve this through synaptic downscaling, but slow wave activity, which is the marker that we usually use for this, is you can only measure during non-REM sleep. So it kind of leaves us with the question of how, or if there is a different marker, one that is not just present in non-REM sleep, can be used to track plasticity and its burden as a continuous process. So we did a study where we had 29 participants complete 3 conditions. So they had an overnight learning night, a sham control night, and then a wake learning session. And each had a matched encoding, immediate recall, and delayed recall task. So we did a word pair learning task and a sham word pair task. We used high density EEG continuously throughout pre-sleep, wake, overnight sleep, and the subsequent morning. So we used a 128 channel electrode cap. We used spectral parameterization to separate these rhythmic oscillations from our continuous background signal called the aperiodic or the 1F slope. So a flatter 1F slope kind of means a more excitable cortex, and then a steeper 1F slope means a little more inhibited. And importantly, unlike sleep spindles or slow oscillations, this signal exists in every brain state, so we can track it continuously during sleep and wake. So We found that during pre-sleep wakefulness, declarative learning produced a significant flattening of the 1F slope compared to our sham control section. So right after learning, the cortex kind of sits in a more excitable, more desynchronized state than it does after the control task. So then continuing through the night, during the first 30 minutes of non-REM sleep, we found that's the window of like maximal homeostatic pressure. That pattern completely inverted. So the slope becomes significantly steeper following learning compared to the sham task. So the flattening from the wake session doesn't just fade, it kind of actively overshoots in the opposite direction once sleep begins. So this is kind of suggesting that sleep as a whole is kind of nudging the cortex to a more excitable state. Spatially, this was not a global effect. So my friendly topo map here shows that the flattening of the 1F slope in that pre-sleep wakefulness was concentrated over prefrontal, frontal, and central electrodes. So the regions that were used during learning were the ones that were more renormalized during that subsequent sleep. So individually, that those flattenings and steepenings of the 1F slope related to next day memory retention. So, but in opposite directions. But the contrast between them, like the size of the wake to sleep shift there, was the strongest and most reliable predictor of overnight memory consolidation. And it was explaining more variance than either state alone and was robust after accounting for oscillatory measures. So together, these findings are kind of suggesting that sleep doesn't just support memory through replay overnight, it actively renormalizes the excitability of those specific cortical circuits that the learning dampened. I have the link to my preprint here. Please download my preprint and read it. We're working on publishing some of this stuff now with a little more info about REM sleep, but that is all. Thank you.
Bravo, and right on time. Next up. Oh yeah, you're already sharing. Perfect. Take it away. Are you also unmuted, Jim Bone?
Walk to the podium.
Oh, here you are. Okay, you were there. Okay. Google.
Okay, hello everyone. My name is Junbom Kwan, and I'm pursuing my PhD degree in UT Austin, and I'm working with Dr. Pasley. And our lab has an asset called brainlife.io, where we can upload our data set and analyze our data, and we can keep track of all the analysis pipeline in the platform. So in this talk, I would like to introduce my degree research using AI models to understand the mapping between function and brain, function and anatomy in the brain. So the first project is on track segmentation benchmark for deep learning models. So track segmentation is to get a white matter track from diffusion weighted imaging. These tracks can be used for understanding psychiatric disorders or how our brains connect to each others. So there is a Myson paper called TrackSec, but the biggest limitation in this domain is that there is no benchmark to test generalizability of the track segmentation approaches. Previous benchmark is 105 human cognitive project dataset. To this end, we use brainlife.io to apply the same pre-processing pipeline for getting the white matter tracks for 3 different data sets, Human Connector Project, CanCan, and Ping, to get like 1,500 of samples. Secondly, I worked on visual connectivity and asymmetry. In this work, we understand the functional map pretty well, but the connections in the visual cortex is less known. To this end, we made a pipeline in brainlife.io combining Matrix or FSL kind of pipeline to make to reconstruct visual connectivity. Visual connectivity. So using this connectivity in the visual cortex, we try to see whether the streamline density follows a specific patterns in our perception. For example, our perception has some asymmetry in depending on polar angles, and these streamline density show the same patterns as our perception does. And the last project was on mapping visual function from brain anatomy. So the main question here is, can deep learning models learn individuality of retinotopic maps from brain anatomy? So using the same deep learning models, I tried to predict the retinotopic maps from brain anatomy, like curvature or myelin map. So when I use existing model based on graph neural network, it seemed like it is predicting pretty well compared to the ground truth on the left. And I also added a transformer-based models, and it seemed pretty good as well. But when I compared these outputs with other subjects, it is obvious that graph-based neural network tend to resemble the ground truth, but transformers prediction tend to be same across subjects, which means that these graph neural networks are capturing some unique patterns in individuals. So we need to use these assumptions for making the better architecture. Yeah, that's it. Thank you, everyone.
Fantastic. Next speaker is Lara. Let me find the title, or you'll see it soon on your screen. About what bridges in Königsberg have to do with my brain research.
While I'm setting up, oh, that's probably not what I want to share. Can you see it?
Not yet.
Oh, sorry. Who knows what graph theory is while I'm clicking share and then share.
Approve the sharing, I think.
Oh, open system settings. Okay. Okay, and then I have to.
Okay, she's got to log out and back in because she's changed her settings. So 1 second.
Okay, maybe we will go to the next speaker first.
Back.
Sorry, America Romero, are you there?
Yeah, she's on the way.
All right. Cool.
Okay. Hi everyone. I'll show you some ongoing work that I'm working on. We have an N of 2, but I'll show you some behavioral pilot data. Yeah, if you have any questions about or any ideas on analyses, please let me know. I'm happy to talk more about this. So as we navigate through the world, we encounter highly similar events. So like if you drive into work, you might park in the same lot every day, but high event similarity can cause memory confusion or memory interference. And so interestingly, work has shown that instead of representing these memories as highly similar, the hippocampus can actually drive these representations apart from each other and look very dissimilar, which is pretty counterintuitive. And there's a few factors that might drive this differentiation, but one factor that I'm curious in is visual attention. I'm curious in understanding if visual input, if that's already differentiated on at the outset, or if that's undifferentiated at the outset, if that then impacts the hippocampal representations. And so we created this set of stimuli that are highly controlled. So we'll see, zoom in maybe, but I'll show you some examples. We created these through Midjourney. So there's one quadrant where there's some differences. The rest of the quadrant, there's not any differences. This is what we call a diagnostic quadrant, where there's some differences. And so we have participants study these scenes through an associative memory task. If I can get this to work. But participants study these scenes with an object associate. All of these scenes are split into a diagnostic or non-diagnostic condition. In the diagnostic condition, there's an arrow that precedes the scene that points in that quadrant where there's some differences. And the non-diagnostic condition, the arrow points away from that quadrant. And so participants are asked to memorize these pairs, and they'll be tested later on. Importantly, in the fMRI version, in the exposure and test phase, there's no arrow that's preceding the scene. And then in the test phase, they have 3 objects that they need to choose from, the either target, or competitor, or an unrelated third object.
And so main point you can't really see here, but participants are doing pretty well on the memory task. They're doing better in the diagnostic condition, which makes sense. We're pointing them in the correct direction, and they're creating more interference errors in the non-diagnostic condition. The also interesting manipulation that we have is this guided insight manipulation. So in previous studies from our lab, we know that the round that participants learn to differentiate is really important for hippocampal repulsion. And so what we want to do is also manipulate when participants are learning the differences between the two scenes. So what you really need to know now is that this arrow changes from non-diagnostic to diagnostic back to non-diagnostic. So we're able to compare when visual input is very similar to when it's not very similar again, and understand when hippocampal repulsion is happening. So some behavioral data. We do see that this guided insight round can boost memory performance, which is what we want. And then what I'm really excited about is some eye tracking data. So what we see here is that in the diagnostic condition, participants are looking in the area where they should be, but in the non-diagnostic condition, what's interesting is that they start off looking in the wrong place, which is where we direct them to. But then only after one round do participants are able to pick up those differences and then continue to look there, even though they're being misguided with the arrow back to the non-diagnostic condition. So hopefully in the future, we'll start looking at the fMRI data, look at specific subregions in the hippocampus, and then early visual cortex as well. And there you go. Yeah.
Fantastic. And we will go back to Laura. Are you ready? Yes, you are. Fantastic.
That's okay. So who's working with graph theory? Okay, some people who's working with connectivity. It's basically then you're working with graph theory. So this is more like, for me, an informal gossip session about the lore of graph theory, because I feel like it's a cool thing to know. So in 1739, people in Königsberg had a huge problem. They had 7 bridges in their town, and they wanted to know if they can cross every bridge exactly one time. And well, they couldn't figure out if they could, so they had to do like drastic things. They had to call the Avengers. And back then, the Avengers were basically Leonhard Euler. Leonhard Euler was an extremely influential mathematician. I think he still is. I don't know if you know the Euler number. I know the Euler number. I should know it. I actually don't know it because I skipped so many math classes, but I know that it's important. So they texted Euler and they said, can we cross every bridge in our town exactly one time? And Euler was like, what the hell? I'm a famous mathematician. Like, what do you want? That's not even mathematics. That's your problem. But then he thought about it and he was like, wait a minute, it's actually a mathematical problem. Because this information on that map is actually mostly not important to us. Because basically all we need to know is that there are 4 landmasses, which he calls nodes, which are connected via 7 bridges, which he calls edges. So all we need to know is that there are 4 nodes, 7 edges. And then he says we can solve this problem by calculating the degree of the node, which is basically how many edges, how many bridges are connected to a node, to a landmass. And it's, I think, very logical. If I want to get on the landmass, I need to cross 1 bridge. And then if I want to step away from the landmass, I need to cross a 2nd bridge. So the number of bridges needs to be even for the question of the Königsberg people to be answered with yes. And in a case where you can answer this question with yes, he calls this an Euler path. So he just named it after himself. I would do the same thing if I would be that smart. But let me see, it's actually, can I use my pen? A famous German example, I think it's a German example of an Euler path would be I can't use the pen function. I don't know if you know this house. We call it the house of the Nikolaus. You basically have to paint a house and you're not allowed to lift your pen. And there is only like 2 ways to do it because only the endpoints have uneven node degrees. So the endpoints can be uneven and all the other points need to be even for it to be an Euler path. And Königsberg, unfortunately, is not an Euler path. So that's the answer. That's what he came up with. Let's see, I kind of can't click anymore. What do I have to do to get my...
So if you
Yeah,
click on the
okay.
screen, then you can keep advancing.
On the what?
If you click on the slide, maybe you can keep advancing. There you go.
Okay. So yeah, basically I'm doing the same thing in my research, or lots of us are doing the same thing in our research. We have a bunch of nodes in the brain, which are our brain parcels, which are then connected via edges, which are the correlations between the bold time series of the brain. Yeah, and this was all like founded by Euler. It's actually used in a lot of disciplines, not only neuroscience. For example, there are Shakespeare papers which are analyzed with graph theory, and turns out that the shorter works of Shakespeare are sometimes more complex in terms of the graph of the character relationships. Yeah, I don't know how much time I have left. I can tell you guys some more if I have some more time. If you want to get started with like looking into the theory and understanding it, I recommend the papers from Olaf Sporns, because he explains it in a very great way. And yeah.
You don't have to.
Fantastic. Thank you, Lara. And on to Jillian, who's already sharing the screen there.
On real. Yeah. So,
All right. Hello, everyone. I hope everyone can hear me. So I'm Jillian, and I'm going in a bit of a different route today, talking a little bit about community engaged research. And so I'd like to begin first by starting, as many great talks do, with a quote. And so earlier last week, I saw this quote during Kendrick Kay's academic talk, which said, "Science comm is so important, but I feel that there's not much training on it, especially for early career scholars." I saw this in the slide and I thought, oh, I have a solution to that. So first, I'll give a little bit of background about sort of the theory, what is science communication, and community engaged research. And so according to the CDC, this is the process of working collaboratively with groups of people connected by geography, shared interests, or common experiences, to address issues affecting their well-being. And so basically, I am working with a team to develop a community engaged research program. And part of this includes rejecting the old sort of traditional deficit model, which posits that communities sort of lack the scientific knowledge, and it's a little bit more paternalistic and top-down. And instead, we are accepting the inclusive science communication, or ISC framework, which you can see here. Basically, this highlights three main sort of tenets: intentionality, reciprocity, and reflexivity. And so intentionality is sort of defining values, goals, and like decision rights and tenets of success with community partners from the outset. It's a, from start to finish is science communication. Reciprocity is creating mutual benefit. So providing compensation, sort of authorship opportunities for credit sharing, and capacity building, and also sharing resources as well. And reflexivity, which is practicing structured self-examination, sort of checking in with yourself and saying, being willing to adopt or adapt in response to feedback and changes throughout the project that you might be working on. And so this kind of goes back to the gap and the original quote. So communities do have a real historical reasons to distrust research. You can think about this in terms of Henrietta Lacks or the Tuskegee syphilis study as well. And poorly done engagement is truly tokenistic and can kind of deepen harm. And honestly, researchers, especially young career researchers, don't really get much training on how to do this well. And so that is the goal of our project, is to sort of make this community engaged research, or CER, program, which will be open source, asynchronous. We're starting small, just at UConn, where I am located, and then sort of adapting across institutions and connecting, after completing the modules and our training program, connecting researchers with community resources and partners. And so this is a little bit of background about what our program is. It's 3 modules, 7 short videos. We have 3 modules. So first one is about inclusive recruitment practices. The second one is about accessible science communication. And lastly, it's about building and maintaining community partnerships in the short and long term. And each video comes with real examples from specifically to like sort of communication disorders, which is specifically where my field is, and as well as reflection prompts for researchers to sort of reflect on. And I know I don't have too much time today, but if you're interested in learning a little bit more and seeing what sort of resources we have and the specific details and outlines of our training modules and what they contain, feel free to scan the QR code on the screen. Thank you.
That was great. Thank you so much for that. Julian, and now on to Tamaya's talk. All right, take it away.
Can you see my screen and everything?
Yeah, looks great.
Okay, perfect. So, hi everyone. My name is Tamaya Levy. I am a third-year PhD student at the University of Oregon, and I'm in the linguistics department. So today I'm presenting an overview of some collaborative work between a couple of departments at UO on testing the reliability of civil clip electrodes. So first, I want to identify the problem. And the problem is that the standard 64 electrode design makes it sometimes difficult to use with certain hairstyles, such as locks, like I have, or box braids, and with certain hair types, such as coarse, dense, and curly hair. So when you think about it, these portions of the population have been underrepresented in EEG research. And so this can mean that medical care is less reliable or accessible for these groups. So we have an inclusivity problem, but potentially a clinical one as well when we think about how are we diagnosing these populations with the tools that we have. And so with this in mind, as a solution, other researchers at Carnegie Mellon University created the Sevo clips. So the Sevo clips are here in the left of the screen, and this is a photo of a pilot participant with the clips. They maintain contact and access to the scalp, and they're 3D printed barrette-like electrode holders. And so with our collab, we are figuring out clip placement per hairstyle, per participant, and really documenting how researchers can use the electrodes and focus on participant experiences as well, thinking about comfort when we work with these underrepresented communities. And also how feasible is it to use the clips if we just ask people to come as they are instead of asking them to change their hairstyles, really putting more labor on participants. And so with our study, we're still in data collection and we're testing the signal quality of data acquired between the Sevo clips and to compare it to the standard cap. And we're doing this across a multitude of tasks through event-related potentials and EROs, or event-related oscillations, and resting, high-low close, and button press tasks. So looking at N400, semantic processing and congruency effects, as well as beta synchronization associated with the button press task. And so it's our hope that with the Sevo clips, they will produce comparable signals, demonstrating that they're interchangeable, which will really inform how we design experiments in the future to really flexibly use the different electrodes that we have to accommodate and include more participants in the research that we do. And I just wanted to kind of give an overview of our collaboration, and everyone here is at the University of Oregon. Thank you.
Great work. Great talk. Thank you, Tamara. That was excellent. Next up is Henrik. Are you ready to present? Oh, I can see that Henrik is trying to share his screen there with help from Noah. There we go.
Okay.
You got it.
Okay. Yeah, so my name is Henrik. I'm from the University of Oslo. I made a webpage in June that is called openmritools.com. I've had a very active summer where I've been attending a lot of conferences, a lot of different places, hearing about a lot of tools. And they all seem very useful. And most of them start with neuro something or end with neuro something. And I find it very hard to know when to use them and to remember them. And my notes are full of tools I should use and stay updated on. So that's why I made this page. It's supposed to be simple and non-bloat. It's just the name of it, whether it's actively maintained. So it's like a GitHub action that runs once a week. You can sort it by like GitHub stars, programming language, and also the modality that you're working with. So it's quite simple. I'm going to rush through the page because you could see that for yourself. There's also a lot of like useful stuff that I find useful, and hopefully others as well. And yeah, I mentioned the GitHub actions. This is how you contribute, and it's just trying to be minimalistic. So you can go in very quickly, flag and edit, or suggest a tool, or you could also fork the page and contribute yourself. So yeah, and this is where it goes. You can see what happens when you contribute. And let's see. Oh, and it started as my project in June, but I hope it doesn't continue that way. It would be great if I'm not the only person trying to stay updated on this. It would be so much cooler if we're, I don't know, 60, 70, 80 people in here that all add just one tool. And also, as a small price for contributing, you get your GitHub name on the on the page. And if you need anyone's permission to put this on your CV, then you got mine. Also, I take the responsibility of maintaining, like actively maintaining the OpenMRI tools page, but we also have openimagingtools.com, which is completely empty and could be taken by any of you. I've paid for a domain for 1 year, and as long as it's going to be updated and maintained by someone, I'm going to continue paying for it. I think that's it, and I just added the next presentation as well. So please come find me and talk to me if you're curious about any of it. I hope that this will be a community-driven project and not only by me. Okay, thank you.
Nicely done. Next up is Federico, who will talk about, I'm looking for it, introducing the needle library. Intrigued. All right, take it away.
Yes. Hi everyone, I'm Federico. I'm about to start my PhD in neurodegenerative disorders and psychiatric disorder modeling using neural networks in the Gaia lab at the Neurospin Research Center in France. And today I will introduce you Needle, which is a Python library developed to make deep learning, and in particular self-supervised learning, easy and fast to access on neuroimaging data. Needle is developed by Benoît Dufumier and the rest of the Gaia lab, including myself. And our goal with this library is really to solve two of the main limitations that limit the applicability of deep learning on neuroimaging data. The first one is that we don't have many high-quality annotated data. And the second one is that getting deep learning to work in a fast and efficient way is quite complicated if you don't have a computer science or mathematics background. So Needle tries to face both of them by allowing self-supervised learning, which means that you train on unlabeled data. You can extract knowledge from this kind of data that is usually much more abundant. And at the same time, we give you an implementation of everything you might need to build a deep learning pipeline so that you, researchers in general, can focus on high-level reasoning and posing the right research question rather than battling low-level, complicated implementation details. The way we do this is with an interface that you can see on the left that sort of resembles what we have in scikit-learn and NileLearn, so that we favor an easy transition for everyone that already knows these tools. We have implementation for the main self-supervised learning strategies, for the main types of neural networks adapted to 3D volumes, and also for data loaders and preprocessing pipeline for all the main dataset of the neuroimaging space. In short, what Needle gives you is an efficient implementation of all the bricks you might need to build a deep learning experiment, so that everything you have to do, if you want to use deep learning in your own research, is just sticking these bricks together in a way that is meaningful for you, for the goals you are trying to achieve. And on the right, you can see an example of code that sort of does this. It implements the training of a ResNet model, which is an 11 million parameter model using contrastive learning on a data set that has 5Ks of 3D volumes unannotated. This is usually a complicated experiment to get to work at first attempt, at first sight, especially if you are not familiar with neural networks. With SimClear, with Needle, you can just do this in like 5 lines of code. So that again, all your work goes into getting right the experiment, getting right the details of the experiment rather than the implementation details. As a final note, this is very much in its infancy, so any sort of contribution on what we might add to the library and everything that we might have done wrong is kindly welcome. And thank you, Andrew, for the publicity.
Thank you. Good job.
Who's next here?
Great talk. And next up is Siddharth. Looks like you're approaching the podium.
Okay. Continue to share. Okay. Yeah. So good afternoon now. I will try to keep it short. So I'm interested in a bunch of things, but this is something also I'm in addition interested in. So knowing about the neural mechanisms of auditory attention. So because just as we are speaking and listening to different talks in this room, we're executing different types of spatial attention. Some of us are paying attention, some differently, and some of us are trying to pay attention but not able to. So these all mechanisms are guided by our brain and slightly different processes. So what happens? I would like to try to understand. So basically, auditory attention, I believe, shapes large-scale brain networks. And my working hypothesis is that task-relevant auditory stimuli will produce stronger activity in salience networks as opposed and stronger and at the same time will have stronger deactivations in default mode networks. And this will vary both by attentional stage as well as developmental stage. By developmental stage, I mean it will vary differently in children as well in adults and younger adults and in older adults. As the circuits are maturing and these balance, as you can see in the green here, would differentiate. So to try to understand this better, this is from ChatGPT. No, I'm just putting it out there. So we all do GLM, right? Once we do GLM, we see activity in the brain in some parts of the regions. And then what I'm trying to do here is then taking those activity and summarizing into on top of a brain atlas. Then we can get average activity that can be then modeled in terms of extent or strength within a network. So in this case, I would be looking at salience network and the default mode network. So the task I'm using is actually auditory attention task, auditory audible task where participants are listening to targets and non-attending to non-targets and deviants. So when they do that, you can see it's beautifully that in the targets, you see higher activations within the salience network. This is across subjects in the sample of 30 children. At the same time, there's higher deactivations also within the target sounds. And for non-targets and deviants, it is less. So I hope I'm able to convince you a little bit that the context of the attention matters. So, and targets evoke greater activity. Yeah, thank you.
Oh, sorry. Jesse, are you there in the room or are you on Zoom?
Approaching.
Oh, you're here. Okay, I can. It's hard to tell from here.
Okay, is this the best angle for the owl?
Oh, face us a little bit on the other side, just so you're.
Okay. Okay, hi everyone. My name is Jessie, and today I'm going to be talking a little bit about my work in the BOSU lab at NYU, where I was using optogenetics to study spatial memory in mouse hippocampus. Okay, so my lab studies the circuit mechanisms of learning and memory, and in animal research, we study these processes using spatial navigation because of this property in the hippocampus where pyramidal neurons become spatially tuned or selectively active at certain locations in an environment. And when hundreds of pyramidal neurons do this at the same time, it creates this unique population spatial map that we know from previous research is generally stable in an unchanging environment, but the tuning properties of the neurons can remap and are plastic in a changing environment. So if you think about it, both of these processes are really important for an individual to both navigate a familiar environment, but also to adapt to a changing one. So, and then I guess the other player you need to know about is the entorhinal cortex, which is the main cortical partner of the hippocampus. They send dense tracts of axons to each other. And in my project, I was investigating how specific subpopulations of neurons in entorhinal cortex shape these properties of stability and remapping in the hippocampus. And then just a little bit about my methods to do this. I used viral vectors, in this case to express like an inhibitory opsin in the entorhinal cortex, and so that I could suppress their activity in their inputs to the hippocampus at certain time points. And then I also express a calcium indicator called GCamp in the hippocampus, so I can record cool recordings like this. So here I'm performing 2-photon calcium imaging of this field of view where you can see a couple hundred neurons in hippocampus with single cell resolution while the mouse is awake and behaving. Here it's just navigating a circular track. So with these methods, I can then extract, kind of like how you guys have like a bold time series, I extract fluorescence over time for each neuron as the raw data, and then we can do some cool basic things with it. For example, if we bin the fluorescence for each neuron by position, we can create tuning curves, rough tuning curve estimates. So here we can see for about like 100 neurons, they each have a distinct tuning center, and together as a population, they tile the entire length of the belt. So we can actually decode with some accuracy where the mouse is at any point just from mineral activity. Also, if we like do some PCA, collapse about 600 neurons onto 2 dimensions and plot it, post hoc color by position, we see that in hippocampus. Again, there's this intrinsic spatial organization to the activity. Okay, so now that you understand the method and the data a little bit, I'll show you some like brief overview of my results. So earlier I was talking about this concept of stability and remapping. So here's an example of what I mean. So this is the tuning curve. Okay, it's a bad example because there's actually 2 centers, but this is tuning curves for 1 neuron in repeated exposures to the same environment. So you can see in exposure A, there's 2 tuning centers, and then in A2, which is a separate recording session, the tuning centers for this particular neuron are stable. They're in the same location. However, and this is kind of a spoiler, in the condition where I'm silencing a subset of the inputs from entorhinal cortex, we can see that for this neuron in session A has a particular tuning center, but it completely remaps in A2. So if we would take the dot product of these 2 vectors, it's 0, and we'd say this neuron is very unstable. So if we take some of these concepts and quantifying stability out of 1 neuron and go to the entire population, we can look at this, we can quantify like the population vector correlation, or how correlated the population is in each spatial bin in repeated exposures to the same environment. So on the left, usually, and this matches what we know from the literature, in the control condition, each location on the treadmill is highly correlated with itself in repeated exposures. But when I do the silencing, I see that that nice diagonal we have in the control has degraded pretty significantly. It actually is significant across mice. And so with this research, we've found some early evidence that this very sparse subpopulation of neurons in the entorhinal cortex has an outsized effect on the stability of hippocampal spatial representations, which is super cool. And I'm happy to talk more about any of like these methods or results if anyone's curious. But yeah, that's all.
That was great. Very, very compelling results.
Am I right? That's the last one?
I know we have a couple more.
Oh, sorry. Okay.
Two more. Next up is Tian Yang, who might be in the room or on Zoom. Here we go.
Hey, hi everyone. The title is different from what I submitted because I was thinking to redo my presentation that I did at CNS, but I decided to not do that because it's kind of boring. And okay, so where is it? Okay, and when I introduced myself, I said I study the aging brain, but aging brain is a very broad topic, right? And the key concept I study is called neurodifferentiation. So I decided just to give a very brief introduction to neurodifferentiation. So how many people here have heard of neurodifferentiation? Okay, very few. Good. Actually, okay, so I hope what I'm gonna talk will at least orient you to neurodifferentiation. So next time when I introduce myself, I will say I study neurodifferentiation. Okay, so here's an overview. So our brain shows different patterns of responses to different types of stimuli. For example, different regions are activated when we view faces as compared to viewing houses, and only a small number of areas that are activated by both are shown in yellow on the right nose image. However, as people age, those yellow areas get larger. That means the new representations get less distinctive with age. That's what we call neurodifferentiation. And neurodifferentiation is associated with cognitive outcomes such as fluid processing. And it's also been established across different modalities such as visual, auditory, and motor. So that's neurodifferentiation at the representation level. And what in this example, what I just showed you is at the univariate level, because the definition of neurodifferentiation can depend on different measurement techniques. And the MVPA is another technique where the differentiation researchers commonly use. So at the MVPA level, it measures this differently by asking how distinctive the overall activity patterns are. So different methods, but the same idea. New representations become less distinct with age. And we can also examine neurodifferentiation at the level of large-scale brain networks. So let's start with what a network is. I'm pretty sure people have some basic ideas, but here's the overview. So at rest, the activity in our brain regions coordinate to form well-organized networks that support specialized and segregated information processing. On this graph, for example, each color represents one specific network. And this structure actually changes with age as within network connectivity decreases while between network connectivity increases. And as you can see here, especially on the graph on the right, so differently colored points become more intermixed, which suggests that the boundaries between those specialized systems are becoming less distinct. In this case, the segregated networks become less segregated, aka desegregation. So that's the differentiation at the network level. Sounds pretty familiar, right? Because across both levels, the differentiation describes the same basic phenomena. Previously distinct patterns of brain activity becomes more similar. And based on that, there are many interesting research questions we ask, such as why the differentiation happens, Like when I was introducing myself, I said I'm also interested in the GABAergic system and cholinergic system, and these are the potential mechanisms that we hypothesize. I have presentations on GABA and acetylcholine and their relationships to the differentiation. So if you're interested in that, I'm happy to share with them. I remember I presented the cholinergic project at SFN last year, and Ariel came to my poster and asked me questions about that, which I was very inspired by that. And we're also interested in how does the differentiation lead to various cognitive outcomes. So we also have a longitudinal data set investigating whether it's age-related differences or actually aging. So that's it. I hope my presentation introduced you to the differentiation. And if you have any questions, please come talk to me. Thank you.
Great, super interesting. And we do have one more talk. Our last talk today will be given by Anisha, I believe, and has a great title, so I'm intrigued. It's called Busy Beavers. Oh, just what we need, pictures of beavers.
All right, this was a bit of a last-minute addition to the program, so thank you for accommodating me. But hello, everybody on Zoom and in person. I am Amisha. I am a second-year PhD student at Columbia University. And most times I spend my time thinking and talking about the neuroscience of social connection. But today we're taking a little bit of a lighthearted tangent to talk about busy beavers. And we have been busy beavers ourselves for the past few days, so I wanted to take a detour and share some fun facts that I love about North America's largest rodent, the original busy beaver. All right, to give you a sense of their size, adult beavers weigh from about 35 to 60 pounds, but the heaviest beaver ever recorded weighed about 110 pounds, not too far from me, or 50 kilos. And their beaver job and the singular purpose of their life, really, is to instinctively create dams to stop flowing water. They get their food and materials to build dams by using nothing but their large teeth to cut down large trees. And their teeth, funny enough, never stop growing throughout their entire life, which is helpful when chomping trees is your full-time job. And my favorite thing about beavers, and really the most endearing thing I find about them, is the impact they have on those others around them. Their dams can transform entire ecosystems and allow for other wildlife to flourish, like fish, amphibians, birds, and other reptiles. And they also help prevent wildfires. The world's largest dam made by a beaver is located in Canada in the Wood Buffalo National Park and was discovered in 2007 by an ecologist looking at satellite images. It spans about half a mile and has led to the creation of a 17-acre lake. I don't think I can even visualize that. All right, and since beavers spend their time both on land and water, they are excellent swimmers and can remain submerged underwater for as long as 20 minutes. I think I can last one. Their flat tail is their most distinctive feature, and it also helps them swim and balance on land when they're cutting up trees. And when they're swimming, they also use their tail to splash water and warn off predators and people who get too close, like myself. It happened to me once, but I like to think that they were just saying hello. And although I did meet somebody in New Hampshire who somehow managed to get slapped by a really chunky beaver's tail and ended up with an injured leg. And I still wonder to this day what he did to deserve it. And lastly, they also form complex social structures where they take care of multiple broods. So the beavers have siblings to hang out with. They live together in a lodge that they build where they play with each other and learn how to be good beavers. Once a beaver reaches the age of 2, they usually leave the colony to find a mate and build a colony of their own. And unlike my ex-boyfriend, beavers are extremely loyal partners and form lasting monogamous bonds I hope this is getting recorded and you can watch it. All right. And a beaver lodge, like the one pictured right here, can look like a blob of sticks from the outside, but it's actually very sophisticated with multiple entrances and rooms. They have separate rooms where they clean themselves, feed, and another higher up, which is reserved for sleeping and grooming their kits. Their babies are called kits, and their bedding of grasses and wood chips are changed regularly, so they're quite disciplined. And their walls are made of mud for insulation during the winters. And just like these busy beavers that we just learned about, all of us have been relentlessly working towards our hackathon projects, and here's to hoping that they turn out to be as well engineered as a beaver's dam. And if you would like to go see a beaver dam, about 20 minutes from here after today's sessions are over, please message me because I have located a dam. Thank you.
Thank you. As a recent Canadian transplant, I particularly appreciated your education.
Okay. Yeah.
And
Okay,
we're
so
right on time.
oh, go ahead.